While I have you, a question that our audience has submitted. Uh, we want to talk a little bit about test automation and the analytical challenges that occur with test automation to bring down the cost overall uh, of manufacturing. In your experience, in your experiences, um, what are the analytical challenges, or the challenges in general to test automation? How are you and your team handling it? Just kind of talk us through your experience with test automation. And Narinder, if you can go first, that would be great.
Yeah, sure. I think in some ways, if you think about the rapid sterility processes that have come on, in past 5 to 10 years, and actually rapidly evolving, field where companies are going with really, really innovative technologies there. So that's first step. I think is finding a process that can be finding an analytical method that is comparable and can be validated, and then reduce the overall process times. And then automation in this case is a complete solution being provided by the vendor that can minimize any unnecessary repetitive tasks. But then are there ways to put that more in line? I think what we are right now thinking about is some of the, for example, the flow technologies that are available are pretty exciting, but they're still offline. Are there ways to integrate them more inline and at-line analytics so that we don't have to send the sample over to the QC lab? We can test it right there on the manufacturing floor, and we can go test multiple batches right there. So that's one thing. And in case of autologous manufacturing, you're making those, and once you've formulated your product and you have DMSO in, you gotta cryopreserve it pretty fast. You can't wait for QC results. So those are the kind of things that we are focused on, and how can we find automation to reduce risk to the process, to through not only open steps but also make the process as robust as possible.
Yeah, sure. Okay. And Craig, I want to give you the opportunity to provide your insight on test automation and the analytical challenges that go along with it, to just bring down the cost overall. So, either from your experience or your experience at BlueRock, what are your thoughts on test automation?
I think that's going to be a key focus to drive costs down. It's one that I don't think there's any reason not to invest in that. And we brought up sterility testing, but also RCL testing is a bottleneck for the vector world since COVID has hit and some of the testing labs focused other things. There's a backlog of all kinds of tests, and I think we're all paying additional money to expedite testing at contract labs every day. So whatever we can do to bring into the clean rooms or bring into in-process testing instead of release testing decreases the risk. So I think that's going to be a major focus for the industry, and definitely a focus in BlueRock. I think we focus more on analytical development, preclinical, than I've ever done in my history, because that's the key to actually understanding your product and then showing comparability as you move through various automation steps or process improvement steps.
Sure. Okay. I want to make sure we get to an audience question before we sign off for this particular Cell and Gene Live. We have a question regarding how do you balance development speed versus maturing manufacturing capabilities and industrialization needs? In addition, how do you address regulatory requirements of process change in cell therapies? I would like to pose that question to both of you. And Craig, if you're okay to answer that first, that would be great.
Sure. I think, well, first of all, when I left Biogen to go to cell therapy, I said I wanted to go to a field that CMC made a difference. And maybe I regret that decision, because every day you talk about balancing development speeds and maturing the manufacturer. It is a trade-off. The real answer is many of us have to get the product into the clinic to see if we actually have a product. So you're always going to, at least the early stages, focus on getting a good, high quality cell safely into a clinical trial as a priority. The reason how we're addressing at BlueRock is we actually have a really good relationship with our owners, Bayer, and as we're developing the early stage manufacturing processes, we lean on Bayer to focus on how do you make an industrial scalable piece, because we are having to balance the development versus industrialization. I'm always going to focus on the near-term clinical entry. If you have this leverage where you're able to leverage a partner, as we are internally with Bayer, then they can focus on industrializing across the platform while we concentrate on figuring out how to make the best cell. So that's how we've done it. I think it's pretty unique, and I think that works. The regulatory requirements is difficult because they're still being developed and formed. So I think it's two pieces. Focus on your ability to characterize your product and over focus on analytical development so that you can actually say that you understand your product and not your process. Because we have to break the paradigm that the process of the product is not. You need to be able to have the analytical techniques to define what your product is, and then have a reasonable conversation with the agencies to engage in a meaningful dialogue. And so far, they've been very open for that.
Okay. Narinder, I'd like to pose the same exact question to you. How are you balancing development speed versus maturing manufacturing capabilities? And also, how are you addressing regulatory requirements and the ever evolving and changing regulatory requirements of process change in your cell therapies?
I think Craig said it well there. You will not have a product if you wait too long to bring it to clinic. This is not the industry we are in. Our patients needed these therapies urgently, right? So we have to balance that speed. But at the same time, you cannot take a process to clinic and then find out that you cannot reproducibly make it. You don't have high success rate. You do not have the ability to scale it up beyond. So obviously, those are the considerations. You have to balance that. How can you, if you can make a case for going fast by going a little slow and doing the right homework, or doing it concurrently. One good thing about how the agencies have approached this is they've given us this different avenues. RMAT designation is one where we found it very helpful to have regular, quick conversation with the agencies about how can we, if you're trying to make a change, how can we prove to them that it's safe and efficacious? Remember, regulatory agencies are highly motivated in helping bring these therapies to make them as broadly accessible as possible. These are potentially life-saving, fundamentally transformational technologies. And they know it's going to be hard. Processes where the challenge is going to be. And but at the same time, the primary mandate is safety and efficacy for these drugs. So we have to understand that, and we have to understand our product. I think Craig mentioned about product characterization, developing analytical tools. I would go a step further, understanding deeply what matters for your product. You can measure for cell attributes, you can measure a lot of attributes, but we have often found no correlation with safety or efficacy. So having the data, having that analysis available, that you can convincingly, make the case that a process change may or may not impact certain attributes, and those attributes may or may not have any impact on the product safety or efficacy. We've had good success with some of this. We are being very careful about any changes. Obviously, all of this sounds great, but the last thing you want to do is make changes to a product and then make it less safe or less effective. Nobody wants that. So our primary goal is first making sure you have a strong process that never cannot be you protect the product first.
Sure. Okay. I think we have time for one more question from our audience. How do you see the extent of COGS reduction in cell therapies in the next 3 to 5 years? Craig, I'll start with you with that one.
Sorry, let me take myself off mute. I guess that's a really tough question. I think there's ample opportunity to reduce COGS. I guess it depends where you start from. If you look back from 2 years ago, I think cutting the COGS in half is easily obtainable because of the improvements in vector processes and using dynamic simulations to come up with better labor utilization, et cetera. So I think those things will naturally happen as the industry starts to standardize steps. I think the real question is, how do you cut it in half again? And that really is all about designing processes that are half the time, or as we're talking about universal processes, et cetera. I don't think you're going to see the benefits in the commercial COGS in 3 to 5 years beyond that first 50% reduction, just because it takes a while to get through the regulatory approvals, et cetera. But I would be shocked if it doesn't happen in the short-term years to come from that. And then I do think allogeneic is having a lot of good data recently. So I think, by the end of the decade, we're going to see some substantial improvements.
Okay. Narinder, before we wrap up, I'd love to hear your thoughts on the very foreseeable future and the extent of COGS reduction in cell therapy, and what are your thoughts on that?
I think cost of goods reduction is one thing, but the more important thing here is that can we even make these therapies? Can we even make them available, right? Reagents are very expensive. Vector, we just talked about. Part of the reason vector is expensive is because there's so hard to make and so little that you get. So improving those yields are not only going to make more vector available, more reagents available, but also reduce the cost, right, in that sense. But this is a long haul. If you think about where the biologics industry was back in 90s, we all thought that we're going to see dramatic improvements, and we did see them. But by the time we could fully realize those improvements, it's been 20, 25 years. So I do believe this is something we need to relentlessly focus on. Find all these things that Craig is mentioning, allogeneic platforms, improving the vector yields, improving the reagents, having second sources available, improving plant capacity, utilizing automation smartly to reduce cycle times. All of those things will long-term help us through the cost.
Okay, good. All right. Well, thank you for your valuable insight to the audience questions as well as to our entire discussion today. And that's a wrap for this Cell and Gene Live. Thank you to you, our audience, for tuning in, to BlueRock Therapeutics' Craig Beasley, and to Arcellx's Narinder Singh, and of course to Thermo Fisher Scientific for partnering with us on today's event. This event will be available in its entirety in the next business day or two, so please do check out Cell and Gene at www.cellandgene.com. Check it regularly, but also for this event, and as well as Cell and Gene, the podcast. And we will talk to you soon.